Hi class, this is Dr. I, and I'm going to cover chapter 10, blood, I'm going to try to do this in about 20 minutes. Okay, so I'm assuming at this point you've already read the chapter, you have a decent understanding of everything, and so I'm going to highlight the important aspects of this chapter and give you a little bit of extra stuff here and there. Okay, so here we go. So the first slide here, this is talking about the basic components of blood, and we basically have two divisions here. We have a liquid division, which we call the plasma, and we have what we call the formed elements, or just the cells. Okay, so we got a 55-45 split there. First, let's talk about the plasma. All right, so we'll just go in order here. So understand it's about mostly water, 90% water. Okay, it carries all of our electrolytes. It carries all of our essentials. So think of the stuff from the nervous system: the potassium, the sodium, the calcium. Okay, and then it also carries other things that we need, like chloride and bicarbonate for buffering and whatnot. Okay, so on top of that, we got to deal with the pH of the blood, right? So the pH of the blood is about 7.35 to 7.45, so it's slightly alkaline. All right. And then on top of that, so in addition to the soluble salts, we have a bunch of proteins. Okay, so plasma proteins are called plasma proteins as opposed to proteins that you would be eating as a food source. Okay, and one of the most common is albumin. Okay, albumin is the same protein that we see in egg white. So when you crack an egg open, it's all goopy. That's a bunch of albumin. All right, albumin is also in our blood, and one of the major things it does is it helps with osmotic balance. What that means is it ensures that the blood is always hypertonic, and so it's constantly getting water and being filled. It's increasing the blood volume, and by having a good blood volume, it makes sure that the heart is pumping the blood and it's being efficient. All right, so there's your albumin. Fibrinogen is normally soluble in water. Okay, so think about soluble fiber. If you have a dietary supplement where you sprinkle some white powder into some water and you stir it and it dissolves. Fibrinogen is like that. It's normally dissolved. Okay, however, on occasion, it will precipitate out of solution in response to a wound. Okay, so this is part of our clotting response. So the fibrinogen and the platelets, which we'll talk about later, are involved in coagulation. All right, and then lastly, we have what are called globulins, or immunoglobulins, or IGs, so IGs. So we have IgE and IgA and IgM, and so you don't have to worry about any of that. Those are immune proteins. Okay, so these are antibodies, and basically an antibody is a Y-shaped protein that has gloppy ends that stick to something specific. Okay, it's usually a small protein of some kind or another. All right, so here's the thing that an antibody binds to, here's a word that we'll use later when we do blood type. It's called an antigen. Okay, so the word antigen means any very small protein, maybe 12 to 15 amino acids long, that elicits an immune response, that causes our immune system to wake up. Okay, so that's what an antigen is. Okay. All right, so there's the proteins in the blood. And then lastly, there's some various substances in the blood. So for example, since we're breathing and we're metabolizing and just always living here, maintaining homeostasis, there's going to be waste products produced all the time. Okay, we're going to be constantly giving our cells nutrients, and we're going to always be exchanging respiratory gases, right? And so the oxygen's coming into the blood and the carbon dioxide is leaving the blood. And then lastly, and this touches on the endocrine system we just finished, the hormones are everywhere in our blood, right? Remember, the hormones travel the entire body through the blood system, but they only affect those cells that have target receptors on them, right? Okay, so that's the plasma. Now let's talk about the cells of the blood, the formed elements. So the cells of the blood include two fractions. There's the white blood cells and there are the red blood cells. Okay? So first let's talk about the red blood cells. Red blood cells are anuclear, meaning they don't have a nucleus. So normally a mature red blood cell will basically they're a big bag of hemoglobin. Okay, they're a big bag of hemoglobin, HB. And hemoglobin, if you remember, is a protein that's got 4 parts. It's got 2 alpha and 2 beta chains called alpha globulin and beta globulin. And each one of these chains in the center of it contains an iron. Oh, that's weird. An iron ion, Fe2+. And this is what attracts oxygen. Okay? And so the red blood cells carry oxygen to our various organs by allowing the oxygen to hitch a ride and then eventually fall off and diffuse into the tissue where it's needed. All right, so that's a red blood cell. They're called erythrocytes. Those are the mature red blood cells. All right, and as we'll learn a little later, erythrocytes start out as a more immature cell called a reticulocyte. Okay, so reticulocytes have some organelles still. They have an endoplasmic reticulum, which is why they're called reticulocytes, but eventually they eject it. And so they eventually grow up to become what's called an erythrocyte. So erythrocytes have no organelles, not even a rough ER. They just have, they're just chock full of hemoglobin, basically. Okay? And they have a lifespan of about 100 to 120 days, give or take. All right, let's talk about the white blood cells now, the leukocytes. Leukocytes, even though they consist of the smallest fraction of the blood, they're arguably one of the most important in terms of our long-term lifespan, in terms of our immune system. All right, and so these include things like lymphocytes, monocytes. Okay, so these two kinds of cells here. And then there's three other kinds of cells that, if you notice, they all have in common these little dots. Okay, so these are called collectively the granulocytes. Granulocytes are granular. They have granular material in their cytoplasm. In contrast, the lymphocytes and monocytes, those are called agranulocytes. Okay, because they don't have the granules. All right, let's talk about the agranulocytes first. So lymphocytes, these are our true immune cells. These are our cells like our B cells and our T cells. These are cells that are involved in generating the globulins, the immunoglobulins, the antibodies. Okay, and we find these a lot in our lymph nodes. So our lymph nodes are hanging out, and the lymph is cruising through the lymph nodes, and it's basically being surveyed constantly. All right, we'll talk more about that when we do the immune system later in the semester. Monocytes are cells that are like little Pac-Mans. They eat other things. So they eat your bacteria. They'll take out viral particles. And so they're called phagocytes. They're a kind of phagocyte. Remember, phagocytosis is cellular eating, so we have phagocytes. Now, monocytes that are cruising through the blood, sometimes they infiltrate into tissue, and then we call them macrophages or macrophages. Okay, so a macrophage is just a monocyte that's found its way into some tissue. Okay? All right, neutrophils, they're the most common of all the white blood cells. And neutrophils are involved in your typical everyday eating or destroying of, taking care of bacterial cells and virus particles. So neutrophils neutralize the enemy. That's one way to think of it. Okay? Eosinophils are special cells that they're not as common as neutrophils, and their job is to take out things like little parasitic worms in our blood. Actually, I saw a video the other day. Let me see if I can show that to you. So yeah, so this is a video that shows a nematode, which is a microscopic worm, in what I'm guessing is some kind of mammalian white blood cells, some eosinophils, perhaps humans. Actually, no, here we go. They are human, based on the title here. Now watch this. So each one of these individual cells are eosinophils. You can see them collecting along the length of the worm, just attacking it. Okay, so this is one of the ways our immune system takes out the enemy. Okay, so these are eosinophils. Okay, so those are the wormy. That was the worm video you just saw. All right, and then the rarest of them all are the basophils. And basophils are the rarest of the cells, and these are the ones that help us mount inflammatory reactions. So they contain things like histamines that cause blood flow to increase into local areas. Okay, so these are the cells that cause inflammation. That's the simplest way to think of it. Okay, then lastly are the platelets. Platelets aren't cells, they're actually chunks of other cells. All right, so there's the parent cell of a platelet is called a megakaryocyte. Okay, megakaryocytes are these big multinucleated cells, and they fall into pieces when they grow up, and they form platelets. Okay, and there's tons of platelets everywhere. They're the most common non-red blood cell component of the formed elements, and these are essential for clotting of blood. Okay, so when you get a cut, the platelets are the first to arrive at the wound, and they gather and they work together with fibrinogen, and they form a clot. Okay. So that's the basics of blood. And you can see here a picture taken from just a normal light microscope with some staining. The light areas here, these are all red blood cells, and you can see that relatively speaking, there's plenty more of those than the purplish ones, the lymphocytes. Okay, you can see, I'm sorry, the white blood cells, the leukocytes. So this is a lymphocyte. You can see neutrophils, and you can see platelets in this particular slide. Okay, and notice that the neutrophil here, you can see the little granules inside. So that's why we identify those as granulocytes, right? Okay, I'm actually going to come back to this slide in a moment. We're going to go here now. So this is how this slide talks about hematopoiesis, which is the generation of red blood cells. All right, so the first thing is that red blood cells, I'm sorry, all blood cells are born deep inside bone marrow, okay? And particularly deep inside bone marrow, the red marrow. Okay, so the red bone marrow, that's where blood cells are born. All right, and so there's a stem cell called a hematocytoblast, which is located in the red marrow, and hematocytoblasts can turn into different kinds of cells. Okay, so these are called pluripotent cells. Okay, pluripotent cells are cells that can turn into many kinds of cells, not all kinds of cells. Okay, a cell that can turn into all kinds of cells is called a totipotent cell. So a totipotent cell would be like a zygote, right? Remember, an egg and a sperm fertilize, you get a zygote, and that gives you, a person or whatever. So totipotent cells. Hematocytoblasts are pluripotent. They can turn into many kinds of cells. And there's two basic fundamental kinds of cells they turn into: lymphoid cells and myeloid cells. Okay, lymphoid cells, and as their name suggests, they're in the lymph nodes. Lymphoid cells are the ones that turn into our lymphocytes, our B cells, our T cells, and then really cool cells called natural killer cells. These are the cells that work together to mount immune responses, right? So when you get sick, your immune system, or when you get sick, your lymph nodes swell up, and that's in part because of this vast increase in the generation of B cells. We'll talk about that when we do the immune system later in the semester. Contrast, myeloid cells are the ones that give rise to all the non-lymphocytic cells, right? And so these include the granulocytes, the ones we talked about earlier, and also the monocytes, right? The ones that eat things, in addition to the megakaryocytes, which give rise to the platelets, okay? The erythrocytes come from another cell called a reticulocyte. Okay, and reticulocytes come from myeloid cells. So the myeloid cells have a nucleus, right? They have, myeloid cells have a nucleus. They have the ER, they have the Golgi, etc., etc. When a myeloid cell starts growing up to an erythrocyte, they start casting off all the organelles. Okay, and so a reticulocyte is an immature erythrocyte because they still have some rough endoplasmic reticulum inside them, hence their name reticulocyte. All right, when they eventually eject the rough ER, okay, and by this point they're already in the blood cruising around, so your blood actually has some immature cells in it that are still growing up in addition to the erythrocytes. In contrast, the stem cells are locked away in the red marrow, right? So the reticulocytes are cruising along and they spit out their rough endoplasmic reticulums and they grow up to be an erythrocyte. And these guys have that lifespan of 100 to 120 days, as I said. Okay, so let's talk about, and let's go back a slide here, we're going to talk about what's called sickle cell anemia. Okay, sickle cell anemia is a disease that affects mostly people who live in equatorial regions on the Earth. If this is Earth, people that live in this area of the Earth where it's nice and warm, they have a lot of mosquitoes, right? And mosquitoes carry malaria. Okay, malaria as a disease is characterized by having great amount of fatigue, and eventually you get anemia, and eventually you die. That's the simple version of malaria. It's caused by a very small eukaryotic cell, a protist, if you remember protists from the beginning of the semester. And this protist, part of its life cycle, which is very complicated, lives inside red blood cells. And the simplest way to think of it is they suck up all the oxygen, and that's why they make you sleepy. Okay, it's more complicated than that, but for just a touch and go here, that's the simplest way to think of it. All right, so here's how it normally works. A normal red blood cell, as I said, is filled with hemoglobin, right? And so hemoglobin has if structurally, hemoglobin is 4 total proteins. There's 2 alphas and there's 2 betas. All right. If we look at the beta chain, if we just zoom in here, let's pretend this is beta globin. Okay. There's one amino acid right here that gets mutated. It turns from a glutamic acid, which is soluble in water. That's the key there. It's water soluble. It likes water. It gets, and this is in the DNA, it's GAG. Okay. And that then a mutation happens that turns it into, actually, this is the RNA sequence. So GAG stands for glutamic acid. If you look at the DNA to RNA translation, I'm sorry, the DNA to protein translation, GAG gets mutated to GUG, GUG. So GAG becomes GUG, and GUG stands for valine. Valine is an amino acid that's hydrophobic. It does not like water. And so as a result, we go back up here to hemoglobin beta, the beta chain. That's where the valine is. Okay, so if you, on the outside surface of this protein, if you put a hydrophobic amino acid in there, that's not going to be it's not going to let it be as water soluble. Okay, now here's what happens. When hemoglobin binds to oxygen, it changes shape. Okay, and when it loses oxygen, it changes shape again. Okay, the shape it is in when it doesn't have oxygen. Okay, so in other words, the cell doesn't have oxygen, less oxygen. When its mutated version is there in the absence of oxygen, that causes hemoglobin to misfold. And instead of it being water soluble, it ends up forming filaments of hemoglobin, normally water soluble. If you crack an egg open and you think of all that goopy stuff, that's kind of what hemoglobin's like. It's water soluble. Contrast that to hair. Okay, and that's what ends up happening to hemoglobin. It turns into a filament that is not water soluble, and that filament ends up wrapping itself along the membrane of the cell, contorting it, making it into this sickle shape. Okay, so that's what happens when you have a mutated version of the hemoglobin gene, the beta globin gene. Okay, now here's the thing. You have 2 copies of every single one of your genes, right? You get one from your mom and you get one from your dad. All right, so there's dad, there's mom. Okay, so let's say that your mother gave you a normal, healthy hemoglobin beta gene, and your dad gave you a mutated version of it. Okay, so half your hemoglobin is mutated and half your hemoglobin is not mutated. All things being equal, your blood's going to be fine. It's going to look like that. But here's the thing. When you get malaria, when the malaria parasite delves into this cell and starts living there and using up the oxygen, that starts favoring the cell to look like this. And when the cell looks like that, it doesn't live as long. It's not as favorable a place for malaria to live. And so people that inherit a single copy of the hemoglobin beta gene that's mutated end up surviving malaria better. Okay, so that's called the sickle cell trait, having the trait. Okay, and people with the trait will survive longer. So that means, from an evolutionary perspective, people that live in the equatorial regions on Earth will, on average, have more of the mutated allele in their genome. Okay, because it gives an advantage. Now here's the problem. If you have 2 people, each of them carrying one of the bad alleles. Okay, so say this is a woman who has a bad allele, and this is a man that has a bad allele, and they have children. What are the chances that their child will inherit both copies of the mutated allele? So there's a 50% chance he'll get it from her, and there's a 50% chance he'll get it from dad. And so when you take that into account, 50% times 50%, or 0.5 times 0.5, that equals 0.25. Or there's a 25% chance that an individual born with 2 people that have the trait will actually get the disease. Because here's what happens. If you have no good copies, if you're HB negative, if all of your hemoglobin beta, if your beta globin gene is all mutated, most of your cells are going to look like this. And when your red blood cells are contorted like that, they get stuck in capillary beds. They do all sorts of things. And that's what gives rise to the disease sickle cell anemia. Okay, they're also, people are in chronic pain. They get poor vision. They start going blind. They have kidney issues. It's terrible. Okay, and this is actually one of the candidates for gene therapy using the new CRISPR technology. If you're interested in that kind of stuff, feel free to Google, do a web search for CRISPR plus sickle cell anemia and see what you can find. Okay, all right, so that's sickle cell. And then the last thing we're going to talk about here, well, there's two other quick things we're going to talk about here. This is what happens as part of normal homeostasis. So you can imagine something happens in the environment that results in your blood not being able to carry enough oxygen, and so you're stressed. Okay, so what can cause that situation? Well, severe bleeding, right? You can lose a lot of red blood cells themselves, or perhaps there's some decrease in the amount of hemoglobin you produce. Or physically, there's just not enough oxygen to go around. So in any case, okay, so here's a really neat example. People who do high altitude training, people who are in the Olympics, for example, and they want to sprint really fast, they go up into a mountain, okay, and they run up on the top of the mountains, not literally on top of the mountains, but you know what I mean. There's very little oxygen as we go up in elevation. And so they spend some weeks up here. And what that does is it stresses their body. It causes their kidneys to release a hormone, erythropoietin. Okay, erythropoietin, that's how you say it. So erythropoietin hormone gets released from your kidneys, all right, and your liver to a lesser extent, as it says here. And what this does is it goes and it tells the target cells in your red bone marrow to start dividing. So start making those myeloid cells, and then start creating some reticulocytes that become erythrocytes, right? So basically what this does is it increases your red blood cell count, or it increases your hematocrit, okay? Once you have enough red blood cells to deal with that diminished amount of oxygen, then you re-attain homeostasis again. And so that ends up shutting off the kidneys and the expression of erythropoietin, and therefore you're not constantly churning out new red blood cells. Okay, so that's the basics of homeostasis here. All right, the last thing I want to teach you about is your blood type. Okay, most of us know that we have blood types. You might not know what your blood type is. Okay, but here, so I'm going to give you a whiteboarding here. This is really quick. We're going to talk about A-type blood. We have B-type blood. We have AB type blood, and we have what's called O type blood, right? So what are these things? So here's the easiest way to understand it. A and B, these are proteins that hang out in your red blood cell's membrane. So if this is a red blood cell, okay, there is a protein stuck in here that's A, or there's a protein stuck in here in B, okay? Or it's mutated and you get nothing, which means you have a red blood cell. That's a terrible red blood cell, but this red blood cell has neither of the proteins on it. That's what O is, okay? So O is basically a mutated version of A and B, okay? So again, just like when we talked about hemoglobin and sickle cell, you inherit 2 copies of every gene from mom and dad, right? Or one from mom, one from dad. So let's say that your mom gave you the gene that encodes the protein to make A, and your dad also gave you that. You'd have A-type blood, right? Let's say your mom gave you the A gene and your dad gave you a mutated version of it, so you get nothing. You'd still have A blood, right? Because you'd still produce at least some of the protein. Okay, so using this logic, you can imagine your genotype. This would be genotype. If you remember anything about genetics here, genotype, you have either, it could be AA, it could be AO, you can have AB, and there's your AB type blood, right? Or if you inherit 2 copies of the mutated allele, you have what's called O type blood, right? And so of course the same things here hold true for B. So you can have BB, you can have BA, which this is equivalent to A and B, right? Those are the same. And then you have, you can have BO, There's the smelliest blood type. And then you can have OO, which I already said up here. Right, so basically any combination of, so you have two slots, right? And so the first slot can either be A, or it could be E, B, or it could be nothing. And the second slot could be A, or B, or nothing, okay? And so depending on what's in your genes, you'll produce either some proteins or you won't, or some combination, okay? So when you say you have A-type blood, you're saying you have your red blood cells are covered with this A protein. If you have B-type blood, it's got the B protein. If you have AB-type blood, you'd have both the A protein floating around and the B protein floating around. Okay? All right, so why is this important? What's the big deal? Well, remember, we have an immune system that has to be able to tell self from not self, right? And so we have what are called antibodies, or immunoglobulins, or globulins. And so this is the cartoony version of that. And remember, immunoglobulins bind to something specific. So in this case, let's say that I had an antibody. So this is how you abbreviate antibodies, anti-A. So I had an antibody that recognized on a red blood cell the protein A. Right? You see that? If I had an antibody for A, I would attack any cell that had A. Right? So here's what happens. If you have A-type blood, okay, you have A-type blood, that means your immune system knows A. So it's not going to have any antibodies that attack it, right? You don't want an anti-A antibody if you have an A protein on your cell, because otherwise you'd start lysing your own cells. That would be an autoimmune disease, okay? So somebody that has, okay, so let's go through this now. What you're seeing here is, okay, so the blood serum contains, we're going to pour some person's blood serum on a slide, and that slide is going to contain an antibody that's specific to A, or an antibody specific to B. Okay? So if a person with type AB blood goes through this test, you will see, well, okay, let's step back for a second. So what? What happens if here's a red blood cell that has A on it, okay? And here's the antibody anti-A, okay? So if these react, this is what happens. You end up getting cells clump together, or agglutinate. That's what this is, agglutinate. And it looks like this, okay? This, when you see this, what that means is the cells are bunching up together because there is an antibody that's grabbing them all together and making them pile on. Okay, so this is a positive reaction when you see the clumping. So if you have AB type blood, that blood cell, which has the A protein and the B protein, is going to react with an antibody that has A and an antibody that has B, right? And so you're going to see agglutination in both of these. So this tells you you have AB type blood. Okay, if you just have B type blood, your body isn't going to, your cells will not react with the anti-A antibody. Okay, so again, if this is your blood cell, there's your B, but this here is an antibody to A, there's not going to be any reaction. And so it's going to look like this, no agglutination. In contrast, when you add that anti-B antibody, now you're going to see the agglutination because that antibody is going to come in here and it's going to recognize and bind to your B protein. Okay, and so the same logic can hold for A-type blood, and notice O-type blood. O-type blood doesn't react with anybody. Okay, so that's the ABO blood type as they talk about. All right, now the one thing that this doesn't show you is the RH factor. So let's talk about that for a second. So first of all, RH is called RH because it was found originally in rhesus monkeys, and that's what rhesus is. Okay, so the RH. So it turns out what the RH factor is it's a little membrane protein that ends up transporting ammonia. Okay, and I can't remember if it's in or out, but it's an ammonia transport protein. That's what the RH factor is, RH factor. Okay, all right, and so what RH factor is, either you have the gene or you don't. So you are either RH positive or you are RH negative. All right. So for example, if your mother gave you an Rh positive and your dad gave you an Rh positive, you would say you are Rh positive. If your mom gave you Rh positive and your dad gave you Rh negative, if he didn't give you one, you're still positive, right? Because mom gave you one. The only time you could be Rh negative is if both mom and dad gave you Rh negative. Okay? And why is this important? Well, again, just like with the A and the B proteins, there are antibodies for the Rh, right? So if you don't have the Rh factor, if you're Rh negative, that means your immune system will have an antibody somewhere that binds to it. There's an Rh antibody. Okay? Now, why is this so important? Well, let's say that you have a pregnant woman that has a fetus. This is a terrible fetus, but you get the idea here. Okay, the fetus is Rh positive. Okay, so the fetus across the placental barrier is going to have blood with Rh plus in it. If the woman is Rh negative, she's going to have antibodies that are Rh positive, and so she's essentially going to attack the fetal tissue. Okay, and so that's why this is such a critical thing. And so what we have to do when we have these situations is we basically have to ensure mom here knows what the Rh factor looks like, and so we can inject her essentially with some anti-Rh antibodies, and we can train her immune system to see it and know it. And so eventually, when she finally does have a baby, she could not have to worry about that. Okay, so how did the baby become Rh positive? Well, remember, there's dad. So, so dad, it gave the Rh positive gene. Mom doesn't have it. Okay, and so when they formed a zygote, they became plus minus. Okay, and so as long as you have one copy of the positive, you're Rh positive. Okay, so that's the Rh factor. So all that together, what that means is you can have A-type blood, and you can be Rh positive. You can be A-type blood and be A negative, Rh negative. Okay. You can be B positive, B negative. You can be AB positive, AB negative, and then you can be O positive and O negative. Okay, so those are the basic major human blood types. Okay, now you can understand this. If you're AB positive, that means your immune system has seen everything. Your immune system knows A, it knows B, it knows Rh, and so it's going to be able to see it all, and so you're going to be able to receive any kind of blood. Okay, so that's the universal acceptor. Okay, or recipient. O negative, these are people whose immune system does not have any of the recognition. Okay, and so they can't receive any of these other kinds of blood, but they can donate it to them. And so this is the universal donor. Okay, this is the most important blood type on the planet, because if you have somebody coming into a hospital hemorrhaging after a car accident and you don't know what their blood type is because you can't ask them because they're unconscious and they're not wearing a bracelet that says, hey, I'm AB positive or I'm B negative or whatever, the one blood you can put in their body and know it'll be okay is the O negative. And that's why that's so important. Okay? All right. So that's the end of chapter 10. So good luck with the rest of your studying, and feel free to email me if you have any questions.